Okay, so anyway, I wanted to thank the organizers for this invitation, and it's a pleasure to talk about coevolution and communication of honeybee defensive strategies against Vespa velutina. And here you can see in this image, I think, can you see the red dot as I move it around the screen where I point to things like the bees right now?
Without problem.
Okay, and basically you can see the colony of Apis cerana, and then here it is the hornet Vespa velutina. So next slide. Now, the big picture of my talk is that I'm very interested in information ecology, how information exchange influences ecosystems and communities. And we can think about communication occurring between the bees as they defend themselves against hornets, but also between the hornets and the bees over the course of evolutionary time. And so we can think about the receivers of the signals, the senders of the signals, and how predation, the information provided by predation, ultimately influences foraging, which is very interesting because the foraging of honeybees provides a very important ecosystem service, and that is pollination. But I wanted to talk right now about information flow between predator and prey and how this has shaped signaling. By the way, here you see a nice Apis dorsata, which is also preyed upon by these hornet species. So the work that I'm talking about today occurred in China, in Yunnan province, what you see here in this red dot. And so here is the outline. So I'm talking first about the evolution on predators, then sophisticated signaling, warning and recruitment signaling, and then finally some conclusions. So first, eavesdropping on predators, and here in this image you can see a little bit of the heat ball that forms, and then below that, in this color image, the temperature that's generated in the heat ball. So it's very interesting because in China, these hornets have a long cultural history. And let's see if I make that bigger. Okay, there we go. I think maybe that appears a little bit better to you now. So they're used for food, and they actually have a traditional role in Chinese medicine. So here, this is a bottle of liquor made with the venom of Vespa mandarinia, which is also considered to have an ancient medicinal benefit. So the Chinese have been interested in these hornets for a long time. And here you see my collaborator, Dr. Ken Tan. This is Yannan Chen, who did this work that I'm going to be talking about right now, and Professor Dongshi Hao, who is now a professor at the Institute. And we're here working at Lion Mountain in Yunnan, and we're looking at colonies of hornets that are maintained here in a special hornet apiary. All right, it's not really a hornet apiary, it's a hornet apiary. In any case, these are highly social insects on their own, and we were interested in studying their alarm pheromones. So what we found is that, like many other insects that have stings, the sting venom is associated with alarm pheromone compound. So here you see the PhD work of Yannan Cheng, and her work identified 4 compounds, shown here in color: heptantuone, non-8-N-tuone, non-N-tuone, and undecimtuone, that were key components in the odors of hornet venom. It turns out that here you see in figure A the antennal responses of the hornets in a GC electroantennogram. So basically we're passing these chemical components, and in this case we're looking at it at higher concentrations. As the concentration increases, the hornet antennae, of course, are most sensitive to it. And here we have a bioassay where we have in B the number of hornets attacking a target which is placed on a tree. And what you see is that as the concentration increases of these 3 compounds, heptantuone, non-N-tuone, and undecimtuone, the number of attacks increases. So the hornets are sensing this as a kind of alarm pheromone. Okay, so I mention this because now the question is eavesdropping, and here we see Vespa velutina, and we see it attacking a honeybee, and this is the work of Professor Dong Chihao. And the question is, if the hornet is always attacking the honeybee, can the honeybee evolve to smell the alarm pheromone, the venom of the hornet? And I'm now going to share my screen and show you this YouTube video. So what you're going to see here is what happens when Vespa velutina approaches an Apis cerana colony. And you can see that bees start to land on the hornet, and they start to attack it and form a ball. And now you can see them pumping their abdomens, generating carbon dioxide, and generating heat on that ball. Okay, so I am going to stop the screen share. Okay. By the way, were people able to see the video? Did that work?
Yes.
Okay, thank you. Perfect. So that's the classic heat balling defense. And as you can see here, this is something that occurs in multiple different species, and they're sympatric predators. So we just saw this with Apis cerana and Vespa velutina. We also have it between Apis mellifera cypria and Apis mellifera caussica when they're attacked by Vespa orientalis. We also see it when Apis mellifera ligustica is attacked by Vespa crabo. And we know that Apis mellifera has a very weak defense and no ICU signal in response to the allopatric Vespa velutina, presumably because it has not co-evolved with this particular predator. So our question was, how do these two species, the sympatric Apis cerana that has evolved with the predator and the allopatric Apis mellifera that is not, how do they respond when they're attacked by hornets? So here you can see the hornet alarm pheromone that I discussed. And when the hornet first approaches, the first bee lands and the hornet tries to bite and sting the bee, it produces some hornet alarm pheromone. But after this heat ball forms, and this diagram shows you how we were able to sample odors from within the heat ball onto a spiny fiber, we see that mainly the components of the alarm pheromone that we can detect are honeybee alarm pheromone because they're trying to sting the hornet. And actually in another study we found that the neck area of the hornet, the vera cervix, is susceptible and can be stung by honeybees. Now, this is a bioassay showing the number of heat-balling bees, and you can see it goes up from 0 all the way up to over 50, forming a heat ball. The control is just a dummy hornet with filter paper, no odors. Then we provide a dummy hornet with hornet alarm pheromone. By the way, a dummy hornet is a hornet that's been dried and covered over so that it not have any odor of an actual hornet. And what's interesting here is that Apis cerana actually forms significantly more a heat ball around this treatment than Apis mellifera. So Apis cerana is reacting to the hornet alarm pheromone. We then provide the bee sting pheromone, and interestingly enough, Apis cerana again is attracted, and we know this from other studies, but the response is similar. The similar letters indicate a similar level of effect, no significant difference, to just smelling the hornet alarm pheromone. And then finally, we have the live hornet, which elicits the strongest response of all. And the live hornet is the only response that actually creates a kind of response in Apis mellifera. But as you can see, it's very small, and they cannot effectively kill the hornet or deter it with this very small heat ball. So, knowing the hornet alarm pheromone, we provided synthetic blends of these different compounds and the individual compounds shown here. What you see that for all of these, there really is no response, but when you have a synthetic mixture of the hornet alarm pheromone provided at the correct proportion, then you see that Apis cerana, but not Apis mellifera, successfully forms a heat ball. So again, this is evidence that they are able to detect the alarm pheromone of Vespa velutina. Okay, so why does this occur? I mentioned electroantennograms, and here's an example. So these are traces of antennae. You cut the antennae of bees, either Apis cerana, the first 3 lines here, or Apis mellifera, the bottom 3 lines, and you look and see how they respond to the components of the alarm pheromone as they are passed over the gas chromatograph. So time is showing that the most volatile components are passing first and the less volatile components are coming later. There is a small amount of heptantunone, and there is a tiny response, but it's not very strong, probably because there's a very low amount of this compound. But for 2 of them, nonantuone and undectantunone, you see strong responses in Apis cerana. And in fact, what you can notice here is that there are some unidentified compounds in the hornet alarm pheromone, shown by these peaks here, that also elicit very strong responses in Apis cerana antenna. So it's clear that Apis cerana can detect these components of Apis mellifera, of Vespa velutina alarm pheromone. Now, we're passing this as one hornet equivalent of alarm pheromone. We're dissecting out the sting gland of a hornet and presenting that. Notice here that Apis mellifera really does not have any strong responses at all, again at one hornet equivalent. So it's a nice story in that it seems that Apis cerana can smell the hornet alarm pheromone, but Apis mellifera cannot. Now, we subsequently provided the hornet pheromone at higher concentrations. And if you provide it at 1,000 hornet equivalents, so imagine 1,000 hornets attacking one bee, which is not realistic, then we find that Apis mellifera can smell and detect these alarm components. However, this is not a realistic situation, but it does suggest it has the inherent ability, receptors, to detect these odors. They are just not expressed at a very high concentration. Okay, so in summary here, Apis cerana has a much stronger heat balling response than Apis mellifera. Only Apis cerana heat balls in response to the hornet alarm pheromone, and the Apis mellifera antennae cannot detect, evidently, the hornet alarm pheromone at one hornet equivalent. However, as I said, if you give it 1,000 times that concentration, you do start to see some responses. So it has the potential to likely evolve greater sensitivity to Vespa velutina alarm pheromone. And this likely might explain why in these other species, where they have their sympatric hornet predator, we have the honeybee subspecies being able to respond and form a heat ball. Now, it's only for this species, Apis cerana, that we've actually shown the eavesdropping, but my guess is that something similar likely occurs in other species of honeybees, because it's logical that the honeybee should smell the hornet alarm pheromone. So this brings up to a really interesting evolutionary question, which is, how can you cheat? And here we have images of knights and people fighting. And the idea is that, well, what if the hornets stop stinging? What if they don't use the venom and then the bees can't smell it? But obviously that would be bad for the hornet because the venom is an important weapon of the hornet. Hornets could also escape from this trap by evolving new alarm pheromone compounds, things that maybe the bees can't smell, but hornets can smell. But it's tricky because bees are very good at olfaction, and we would expect them to evolve olfactory detection. So this is a classic example of an arms race where it's very difficult at this point for the hornet and the bee to break out of this situation. So what is going to happen next? Well, on the side of the bee, I want to talk first about what the bees can do to defend themselves against the hornet. I wanted to introduce this concept of the referential communication of danger. So we know that many different animals, some animals that we don't think of as very intelligent, such as chickens, can produce alarm calls. Chicken alarm calls change when the predator is more dangerous. Similarly, Mexican chickadees have higher pitched alarm calls in response to greater danger. Lemurs have increased vocalizations. They do it more often when the danger is more severe. And then social mongoose calls can also vary acoustically. So this kind of referential communication of danger, where greater danger corresponds to a different kind of alarm signal, is quite common. So one question is, does Epicurana use stop signals as a referential alarm call? And a stop signal is a signal that bees typically generate to stop recruitment for a dangerous location. And I'll show you some data on that, but first I wanted to show you, this is Vespa velutina and Epicurana. But look at the size difference between here, Vespa mandarinia, and Vespa velutina. Mandarinia is much larger. So the experiments were to use large and small hornets that we tethered to attack in two different contexts. One, they were attacking foragers trained to a feeder, or they were attacking at a nest entrance and therefore getting heat balled, as you can see here. And this is the work of Professor Dongshi Hao, and he's looking at the behavior inside the nest, what these bees are then doing and signaling. Okay, so first I am going to show you the stop signal. So I'm going to share my screen. Share. And what you're seeing is the microphone. It's a little bit hard to see in the video, but underneath the microphone you will hear the stop signals of the bee. One signal, two, three, four, 5. Okay, so let me stop the share. Okay, so I hope you could all hear that beep sound, and that is the stop signal. You may have been able to see that there was a bee that was waggle dancing, and the reason that the stop signaler was doing that is the stop signals tend to be directed at other bees visiting the dangerous feeder, and this inhibits the waggle dance. The waggle dance eventually stops, and it's stopping the recruitment of the nestmates to that dangerous location. Okay, so how does that look like for the different types of predators? So here on the graph on the left, we have the number of waggle circuits in the control. By the way, these are 3 different locations in the colony. A is the entrance, Z1 is what we call the dance floor, it's where most of the bees are dancing, and Z2 is a little bit further away, there are fewer bees dancing. What you can see that in response to being attacked by a hornet at the feeder, there is a decrease in waggle dancing for that dangerous feeder, which makes sense. The decrease is about the same for the small hornet and the large hornet. But if we measure, and this graph on the right shows this, the number of stop signals, you see that there are a few stop signals being produced all the time, about 4 on average. But when you have the hornet, it now doubles to over 8, and for the small hornet, and with the large hornet, we now have over 20. So colony-wide, there is a very dramatic increase of more than 4-fold the number of stop signals in response to the large hornet attack, about 2-fold in response to the small hornet. And we know the large hornet is more dangerous. It can kill more bees at the feeder when it is attacking. Now, we also did the same thing inside at the nest entrance, and in this case, the bees are coming in and producing stop signals against other bees inside the nest. They're also targeting the foragers, but when the bees are sensing that there's a hornet at the nest entrance, what they are doing is that the bees that receive the signal are behaving a little bit differently. Instead now, they just stay inside. They are inhibited from leaving while the guard bees go out and heat ball and attack the hornet. When they are attacked at a feeder, the stop signals at the feeder cause the bees to stop waggle dancing, but they seem to still go out occasionally and forage and check out the feeder to see if the danger is still there. So it seems like they're responding differently, and we wondered if the signals sounded differently. So here, what we can see is the predator effect on stop signal frequency. We have a sonogram up here, but what we look here is the fundamental frequency of attacking the forager versus attacking the nest. When there's a large hornet, you have a higher pitch frequency. You have that beep sound. And in fact, the video that you just saw were attacks by a large hornet. So that's what a large hornet sound stop signal sounds like. When bees are attacked by a small hornet, whether it's at the nest entrance or at a feeder, they produce a lower frequency. So unfortunately, I can't play it for you right now, but it sounds like beep. So you have large hornet, beep, small hornet, beep, and the pitch changes, as you can see here. The fundamental frequency is decreasing. Now, this is consistent for large hornet and for small hornet. The duration of the beeps change depending upon the context. When the large hornet is attacking the nest entrance, it's much longer. So instead of a short beep for foraging, it's more like beep. And the same thing for the small hornet, it's now a lower pitch, beep, when it's foraging, but at the nest entrance, it's longer. So it's beep. And so in these two ways, adjusting the pitch and the duration of the signal, we think the bees are able to signal the danger and to respond appropriately. So I'm going to skip this slide, and because the next slides I think are going to be more illuminating. One question we had is, can we actually test this behavior with artificial stop signals? So the first thing we did is we used accelerometers and calibrated them with laser vibrometers to measure the vibrations that are produced. And there are two very important numbers here that I want you to see. 9.7 meters per second squared, that is the acceleration. You can think of it as how loud the stop signal is when a bee is attacked by the large hornet. And 5.9 meters per second, which is the loudness, the acceleration of the stop signal in response to the small hornet. As you can see, the large hornet results in stop signals that are nearly twice as loud. And we also wanted to see, and this is a freezing response curve of the bees when vibrations are played back at different frequencies. When a bee gets a stop signal, so the stop signaler comes and she butts her head up against the dancing bee and produces the beep sound. And during the duration of that, the dancer freezes and doesn't move. So what we looked at is the freezing threshold response of these two vibrations. What you can see here is that although bees tend to be more sensitive to vibrations within the range of the stop signal frequencies, this little dip right here, between 250 and 750 hertz, there really isn't very much difference in how sensitive they are to the different frequencies of these stop signals. We thought that maybe they are more sensitive to higher frequency signals, and that's why they tended to stop more and be more likely to be inhibited from waggle dancing. But this graph doesn't suggest that. Instead, we think that there is a far simpler explanation, that the stop signals resulting from the large hornet, which you see here, SSLH, are just louder than the stop signals resulting from the small hornet attacks. So this is a before and after shown in gray, and you can see that the number of waggle dance circuits per dance decreases by half for the small stop signal when the dancer receives that, but it decreases to 1/3, significantly less when the bees are getting the large stops, the large hornet elicited stop signal. And so the large hornet stop signals are significantly more inhibitory than the small hornet stop signals. Now, in a separate experiment here, and here you can see these are the playback wands that we use to play back these signals. We just gave a dancer stop signals until she stopped dancing. Now, she will freeze each time we get a signal, but sometimes she'll continue dancing. And so here, in response to the small hornet, you see that it took about 14 stop signals on average for that hornet to stop dancing. But for the large hornet elicited stop signals, it only took about 4. So they are much more effective at inhibiting the dancing. Okay, so in summary, in terms of this component about referential signals, what we see is that predator danger in general in multiple animal species has led to referential signals. Greater danger in this case of the Vespa velutina and Vespa mandarinia result in changes in signal amplitude, signal pitch. And the receivers respond appropriately to natural and artificial stop signal playbacks. I didn't mention the natural data because it didn't work out in this kind of slide format, but a very similar result. Okay, so now I wanted to go back to the first thought of what happens when you have hornets and you have bees. They co-evolve together. They have this arms race. The bees are great. They have this communication which helps to protect them against the hornet. They also have heat balling, which helps to kill the hornet. So in this kind of system, what you expect is honest communication. You expect that the bees should be able to warn the hornet, there's no point in attacking us because we can kill you. Our colony is too strong. And that's what we're talking about here. This kind of honest signaling can also be called a pursuit deterrent signal. Think about a wolf looking at white-tailed deer and the deer running away and then showing their white tail to indicate that the wolf has been seen. The wolf can actually stop spending energy on chasing the deer because he knows, or the wolf knows, that it doesn't make a difference. It will not catch the deer. And so these are called pursuit deterrent signals, and they're quite common in predator-prey interactions. So for example, Cooper in 2001 showed this in the striped-tailed lizards, and there are many other examples. The second thing that I wanted to talk about is contagious signaling. So imagine you read something on the internet and you just believe it, and you tell your friend on Facebook, for example, and they believe it. And pretty soon, millions of people believe the same thing, but no one has ever checked to see if it's actually true. Now, in contagious signaling, so for example, in these woodlice, this actually can happen, where one woodlouse produces an alarm signal in response to a predator. The behavior and vibrations triggers all of the woodlice to start producing this signal. And it's good because it amplifies defense. Now the entire colony is signaling to the predator that it has been spotted. But as you can imagine, as I mentioned, this problem with news is that we have a runaway signaling problem. Sometimes this is called the boy crying wolf, but we can also think about it as fake news. What happens if members of a collective, of a social group, do not check on the accuracy of the information, but they simply believe an alarm signal? There might be bees that are producing alarm signals by accident. And what can we do about this? So first, bees have an ICU signal, and I will play the signal for you on 1 second. So what you will see here is that we have the hornet presented to the bees. This is in slow motion, and the bees are shaking their abdomens, and they are also flickering their wings. And you can see that this occurs in waves. They start shaking, and then they stop shaking. And then towards the end, you'll see what it looks like in real time. Okay, and so now you see they all start shaking. They start and stop in synchrony. Okay, so let me stop the share and go back here. So it turns out the signals are synchronized, not surprisingly. If you look at the start time of a randomly chosen bee in a group and the start time of the bee of another bee, you'll see that they're very highly synchronized, almost a perfect linear regression, and similarly with the stop time. And what you would see in this video, if it were working, is that the hornet is being swept back and forth, and the bees are all in synchrony producing this ICU signal, not the stop signal. Sorry, I said that incorrectly. The ICU signal. So the idea behind the ICU signal is that the bees are telling the hornet that they see the hornet, and Ken Tan's group has shown that if enough bees do this in synchrony, the hornet will fly away and will no longer attack the colony. So one thing we wanted to know is, can this signal, the ICU signal, attract guard bees? So we have a guard bee on one side of this partition, and there's guard bee on the other side. And as you can see in the diagram, we present the hornet. There's a partition so the bees can't see the hornet. We use live guard bees and live hornets. And we see if this ICU signal attracts the other guard bee, which it does. You can see that whenever there is an ICU signal, the guard bees are attracted, whether or not we're looking at a 2-bee scenario on the left or on the right. And we did this on both sides to eliminate the possibility of side bias. Or if we have a more natural situation where we have multiple groups of bees on both sides, you can see, again, significantly more bees are attracted whenever they see another bee or group of bees producing the ICU signal. By the way, there's a little bit of movement here in the multi-bee experiment because bees are always randomly moving back and forth in front of the colony entrance. Okay, an immediate question is, well, how do we know that the signal is just a visual signal? Do the bees produce alarm pheromone? And so we use the same technique of SPEME to collect odors from around a bee within a millimeter away, producing the ICU signal. And we show that regardless of whether they perform or do not perform the ICU signal, they do not produce any of these major alarm pheromone components. But the alarm pheromone components are definitely present in the bees. These are the sting glands. And this is how we normalized it. All of these responses are normalized to one, the equivalent of sting gland alarm pheromone. And again, if you look here at the analysis here, these are the chromatograms of the odors, what you see is regardless of whether they do not perform the ICU signal or they do perform the ICU signal, there is no significant difference in the odors. So the guard bees are not producing alarm pheromone. But you can always say, well, how do you know? Maybe you're not sensitive enough. Maybe the experiment is not good enough. So we tried a different technique, which is a video playback. So here we have an iPad, a video screen, and I hope you can see there is a bee here on it performing the ICU signal. Unfortunately, I can't show you this video, but what happens is that when the bee performs this ICU signal on the video, no alarm pheromone, no hornet present, the bees here in this group synchronously also produce the ICU signal. And so in this way, we're able to test multiple different visual playbacks to see what visually elicits this signal. So the first thing is, just like in the other experiment with live guards and live hornets, the guards are attracted to the video playbacks. And you can see here significantly more, about 55% of the guards are attracted as compared to 10% of the guards. Well, the control is that we just show the same video, but it's not moving. It's a still image. And so this is simply due to the movement that they see another bee producing the ICU signal. Okay, so now we have this technique and we can use it to test different kinds of video playbacks. So what we have here is we have a butterfly, which is sympatric and is harmless to the bees. We have the bees performing the signal, and then we have hornets. Because we're doing a video playback, we can either do it at half speed, 1 speed, 2 times speed, or even in the case of a hornet, we can do it completely still, a hornet not moving at all. And what you see is the bees never respond with ICU signals to the butterfly. They do respond to a bee, as we showed earlier, but what's interesting is that they only do it when the bee is doing it the right way. The bee has to do it at 1 time speed. If the bee is doing it too slow or too fast, they ignore the bee. They do not produce ICU signals. For the hornet, they ignore the hornet that's not moving. They ignore the hornet that is moving too slowly. But a hornet that's moving at 1 time speed or 2 times speed gets some of the highest responses overall. So the bees broadly are very sensitive to the hornet, but they are very specific when it comes to another bee producing the ICU signal. And this is the idea of contagious signaling. So yes, remember, in this playback, there is no hornet, but all the bees are rushing over to the bee that is producing the signal, in this case, the video signal. And as they start to produce more signals, more bees come in and start rushing out. And so you can imagine this is a case of fake news. And in fact, it is fake news. In this experiment, there is no hornet. But nonetheless, they do have a little bit of specificity. The bee has to do it in the right way. Okay, so in summary, what we've shown in Apis cerana is, first step, a bee sees the hornet. The hornet, this initial bee, does the ICU signal, which visually attracts other guard bees. Oh, and I'm sorry, the last part of the slide didn't show up, but the other guard bees also produce the ICU signal, and if there is enough of them, the hornet will be deterred. And this is why it's so important. The hornet is a major threat, and it needs to be warded off, and it will only do so if it honestly knows that there are enough bees to defend the colony. So a lot of bees have to immediately rush out, confront the hornet, and produce the signal. And doing it synchronously helps to increase the conspicuousness of the signal so the hornet can actually see that there are a lot of bees there defending the nest. Okay, so bees perform the ICU signal to appropriate visual stimuli at the right speed to predators, the hornet, but not the butterfly, and to other bees performing the signal. The signal is contagious. It will start a cascade of bees rushing over to defend the colony against the hornet, and it's synchronized. The ICU signal is not associated with alarm pheromone. It is purely a visual signal, or at least a visual signal alone is enough to elicit the full behavior. It visually attracts guard bees, and as I said, it's a group signal that heightens conspicuousness and honestly communicates the colony fighting ability. So what about the fake news? Well, as you imagine, let's say I'm a bee that sees a hornet, or maybe I don't see a hornet and I make a mistake. I produce the ICU signal. All the other bees, maybe you guys in the audience, are going to rush over to me and immediately start producing the ICU signal at the same time. But you also see whether or not there's a hornet. So I think what will happen is that in the absence of a hornet, if you repeatedly provide the ICU signal from one bee, the other bees will ignore it and no longer produce the signal. So this is one solution to the problem of mistakes. The other bees eventually do see the hornet and will stop signaling, if they do not see a hornet. Another thing we found interesting is that we tried this experiment at different times of year, including times when bees are not attacked by hornets because it's not hornet season. And in those cases, we get very weak responses from the bees. They're not as attracted to the ICU signal. So there may be a seasonal component where the colony is prepared for hornet attacks and is sensitized and therefore is much more likely to respond to these signals. That again can help the colony allocate its work effort appropriately and fight the problem of fake news if it knows at this particular time it's very rare that you would have hornets. Okay, so what's next? We are looking now at other Asian species of bees. This is the dwarf honeybee, Apis florea, and it nests in the open on combs. And we looked at the number of stop signals produced by these bees in response to a small hornet, the weaver ant, or just being pinched at a food source by forceps. And you can see that they also increase the number of stop signals when they are attacked. And so this is interesting. They don't produce an ICU signal because they don't heat ball the hornet. Their only defense against the hornet is that they nest inside very thick foliage. And so it's hard for the hornet to fly through. But if they are attacked, they will eventually abscond and leave their nest behind. But they are able to warn each other using the stop signal. Similarly, Apis dorsata, there's also evidence that it produces a similar kind of stop signal as well when it is attacked by a predator. And this species is a little bit more difficult to study, as you can imagine, so it's something that we're continuing to work on. So in conclusion, I wanted to point out that we've talked a lot about information warfare, that basically the hornets are producing alarm pheromones, the bees are intercepting when they've co-evolved with the hornet the alarm pheromones, and therefore eavesdropping upon these chemical signals. One question is, can they lie to each other? Can a bee colony lie to a hornet and say, I'm stronger than I actually am, and I therefore will repulse the hornet? Well, I suppose a bee colony could, but in the end, the evolutionary honesty is going to be important because the hornets will be selected to realize that the bee colony is bluffing and will therefore still go and attack the colony. So in the end, in the terms of the evolutionarily stable strategy, the bees still need to be able to heat ball and actually defend against the hornet. And I think what's interesting about eavesdropping and this whole system is to think about the evolutionary constraints. Each time the hornet can do something, but the bees can counter that. And they are kind of locked in this battle, as I said, this arms race, where they now have evolved even a communication signal, the ICU signal, that works between bees, but also communicates between bees and hornets. Well, I would like to thank the many collaborators who participated in this work: Ken Tan, Yanang Chen, Dong Shihao, Wen Ping, who did the chemical ecology work, and Yuan Wang and Qi Zhang, who were master's and undergraduate students who worked on this project. And I would like to thank the funding sources from UCSD and the Chinese Academy of Sciences. Thank you very much, and I'm happy to take questions.